All right. Well, thank you for joining us today. We've got a couple of fantastic speakers. And just before we get started, I wanted to remind you to put your questions in the Q&A at the appropriate time, since that's the easiest way for us to deal with questions in this format. So first, today we're going to be hearing from Cheryl Arrowsmith, who is a senior scientist at the Princess Margaret Cancer Centre and a professor in the Department of Medical Biophysics at the University of Toronto, and also the chief scientist of the Structural Genomics Consortium at the University of Toronto. Her research focuses on the structural and chemical biology of chromatin and epigenetic regulatory factors, especially as they relate to cancer and drug discovery. In partnership with major pharmaceutical companies, she leads the Structural Genomic Consortium International Open Science Program that is developing and distributing chemical probes that support the discovery of new medicines. She received her Ph.D. from the University of Toronto and carried out postdoctoral research at Stanford University and was a co-founder of Affinium Pharmaceuticals, which developed a new medicine for multi-drug resistant bacteria. She's published over 300 research articles and was recognized by Clavariate Analytics as being among the world's top 1% of highly cited scientists in many years. And she's also been elected as a triple AS fellow and a fellow of the Royal Society of Canada. So thank you so much for speaking today in this seminar, Cheryl.
Everyone can see my screen.
We can see it. Oh, there we go. Now we have the presenter view.
Okay.
Perfect.
And I'll try to get there we go. Oops, wrong one. Sorry. All right, I'll just use my pointer. Right, so happy to join you all today and tell you about tools for pharmacological modulation of the epigenome and transcriptome. Advancing. Sorry, I'm going to start again. Okay. Epigenetic alterations are common in cancer, and cancer probably relies on these epigenetic changes to both develop and maintain itself. From a therapeutical point of view, the fact that epigenetic changes can potentially be reversed as opposed to driver mutations. This makes this area of interest in drug discovery. There's a lot to learn, and there's a lot to figure out how epigenetics drives cancer and can be harnessed, how vulnerabilities can be harnessed for therapeutic benefit. So what we've been doing for at least 10 years now at the Structural Genomics Consortium is to generate chemical probes, which are drug-like small molecules that selectively inhibit, usually, or at least modulate the activity of specific proteins in cells. And much of work we've been doing has been on epigenetic regulators, although there are many tools beyond epigenetics, including kinases and a lot of other recognized drug targets. So a chemical probe is meant to probe the biochemical function of a protein in cells. It needs to be potent, it needs to be selective relative to other similar proteins, and they need to be able to get into cells. Membrane permeability, for example, and not be generally toxic. So these are the sort of drug-like properties that we try to optimize for. And of course, they're complementary. It's a pharmacological approach that's complementary to knockout or knockdown methods, genetic methods. It's closer to a therapeutic modality than the genetic methods, though, and so it's a great combination. And it's you can use it in temporal fashion and drive acute reduction of a protein or enzyme activity. The compounds, the chemical probes that we generate have been made, many have been made in collaboration with the pharmaceutical industry, but also with several academic chemists who are great collaborators I'll mention along the way. And most of these can be purchased now from vendors and are widely available with no restrictions on use. So we've, in our lab, we've been targeting mostly the methyl, so-called readers and writers of chromatin marks. And colleagues in Stefan Knapp's group have targeted the bromodomains, which are readers of acetyl-lysine. And so together, the protein pockets that recognize acetyl and methyl-lysine have properties that make them amenable to binding drug-like small molecule ligands and therefore fairly druggable. This just summarizes some of the work we've done, both in the Knapp lab on bromodomains and in our group on methyltransferase domains. These two papers summarize a lot of what we've done and describe a lot of the compounds that are available and features of them to help you use them appropriately. But I'll just go over today's a brief summary of the methyltransferase enzymes, and then I'm going to focus in on one set that will sort of set the stage for Riley Horvath's talk, which is the second half of this webinar. So protein methyltransferases come in two flavors, so-called set domain proteins that methylate lysine, and on the left-hand side, phylogenetic tree here, and on the right are arginine methyltransferases that methylate arginine, and arginine can be mono or dimethylation. Dimethylation can be symmetric or asymmetric. Lysine methylation can be mono, di, or trimethylated, and each of these is a different signal. So in terms of pharmacological inhibition, the field first started working on trying to identify compounds that mimicked the cofactor S-adenosylmethionine in this sort of in the same with the same concept as for kinase inhibitors that kind of mimic ATP. And it actually turned out to be hard to do. Only a few of these enzymes could be inhibited readily through the SAM site, and there's chemical reasons for that I won't go into. But more frequently, we were able to identify compounds that bind in the substrate binding site and compete with a histone peptide substrate for binding. And that's where the majority of these types of inhibitors, the mechanism for the majority of these inhibitors. In a couple cases, we've identified allosteric regulators. The arginine methyltransferases are dimeric enzymes, and so these compounds bind at the dimer interface and disrupt activity allosterically. An interesting set are the multi-protein enzymes of the PRC2 complex, where EZH1 and EZH2 are the active enzymes, and the MLL complexes, or SETD1 complexes that methylate H3K4 are also multi-protein complexes, and one can disrupt the protein-protein interactions within them pharmacologically, and then that downregulates the enzyme activity. But what I will focus on today is a new way of targeting the enzyme or downregulating these enzymes, and that is through targeting their reader domain and using the ligand to the reader domain to actually degrade the entire protein, including the catalytic, the catalytically active protein. So we'll get there in a few moments as the story proceeds. And the examples are for the NSD proteins that methylate H3K36. Just a quick note that as I mentioned, the majority of the enzymes we're inhibiting are inhibited through their active site histone peptide binding sites. And because these binding sites tend to be very selective for the peptide sequence that they bind to, we can also exploit that and identify very selective enzymatic inhibitors. So the direct acting inhibitors of the enzyme. This is just shown structurally here where the peptides bind different sequences, and you can exploit the upstream and -1, -2 peptide positions with specific chemical conformations. So in the entire collection so far, we and the wider community have identified inhibitors of methyltransferases that make such that one can modulate most of the major histone methyl marks on H3 and H4, as shown here, and also arginine marks. Turns out that the arginine methyltransferase inhibitors are have much greater effects on some of the RNA binding properties of arginine methyltransferases, and they modify RNA binding proteins, such as those in splicing factors. So the actual epigenetic effects of inhibiting them are probably less prominent than effects on splicing, for example. So this is the collection we have here, and you'll notice there's one site that is missing, and that is K36 here. So all these inhibitors on here right now are direct inhibitors of the catalytic site. We were never able to identify inhibitors that directly inhibit the enzymatic activity of NSD2 and NSD3 or any of the other K36 enzymes. But we were able to identify compounds that bind to their reader domains, which are fall into the PWWP domain class. So this leads me to this topic of how the function of these reader domains and how we can modulate them and how we can use them, exploit them for targeting the entire protein. So in the schematic, we're showing that you have a catalytic domain shown here in pink that will deposit a mark, but where that mark is deposited is usually determined by a number of other factors, including reader domains that are part of the enzyme, illustrated here, and also many collaborating factors as well. And so it's less so the catalytic domain that determines where the mark is deposited, and more so the reader domains and associated factors. And so if we can inhibit these reader domains, you may be able to affect where the marks are deposited in the epigenome. And so this has been tried, intensively worked on. In fact, bromodomains turn out to be quite a druggable protein class, as we know. You're probably familiar with inhibitors of bromodomains of BRD4, for example, or the BET bromodomains that have quite potent biological properties, and were a very popular drug target for quite some time. We've been working on the methyl reader domains, and it turns out they are also druggable. This is a plot of druggability score, but even the less druggable methylysine reader domains, we've been able to identify chemical probes or pharmacological inhibitors too, mostly through work with Lindsay James and Stephen Fry at the University of North Carolina. Chromodomain inhibitor shown here. The PWWP domains I show here. We made a chemical probe with Boehringer Ingelheim and with Lindsay James's lab, and this will be the topic of the next few slides. And then finally, the really druggable domains called WD40 domains are essential subunits of MLL. WDR5 is ML1 is dependent on, and the EED subunit of PRC2. So all of these are reasonably druggable proteins. So now let's focus on the PWWP domain. David Dilworth, a postdoc in the lab, led this study, and he so this structure here shows the histone peptide going from the upper left down across the protein. You can see the methyl lysine group here binding in the so-called aromatic cage, the three cyan-colored aromatic residues that like to form a cage around methyl lysine. And then the lower right of the peptide is actually directly coming out of the gap between the DNA on the nucleosome. And so this lower right side of the protein is really smack up right against the DNA, and also has DNA binding properties. So NSD2, shown over here on the right, selectively binds to the dimethyl mark on lysine 36. And some of the other PWPs recognize trimethyl, sometimes in a histone-specific context, as shown here. It turns out there's there's a large family of PWWPs. And for the rest of this talk, we'll focus on just mostly NSD2. So with Lindsay James's group, she's a medicinal chemist at the University of North Carolina, we developed this chemical probe that inhibits the interaction of the PWWP1 domain of NSD2 with nucleosomes with the H3K36 dimethyl mark. So we have the active compound, UNC 6934, and we always like to have an inactive control, and that's shown here with the only difference between the two molecules is a isopropyl group in the middle instead of a cyclopropyl group, and that completely disrupts the interaction that's buried down in here in the protein. And so we always like to use these together to demonstrate that whatever phenotype we see is dependent on inhibiting NSD2 itself and not some off-target activity that may be unanticipated of this molecule. So we did some work trying to understand the function of PWWP1 domain. This protein has two PWWP domains in it, shown here in the schematic. So the wild type protein has two major isoforms, but in multiple myelomas with a T414 translocation, there are these truncated proteins that lack the PWWP1 domain. And it had already been published that in multiple myeloma, the truncated isoforms tended to have nucleolar localization, not exclusive, but higher nucleolar localization compared to the wild type protein. And we can phenocopy that effect by inhibiting the PWWP1 domain, which ends up disrupted in the mutant proteins. So this was encouraging that we were seeing a mimic of a biological effect already observed. David wanted to understand more about how this works and this concept of whether in a very large protein like NSD2, can you, with all these reader domains, has two PWWPs, a bunch of Ph.D. domains, with a disruption of just one of these domains interactions with chromatin or with a specific mark, could you really relocate it to somewhere else? And so he went through and mutated each of these individual domains with a point mutation that had already been reported to disrupt interaction with histone, including the catalytic domain. And what he found was that mutations in the reader domain would disrupt nucleolar localization. So fibrillary is the marker of the nucleolus. And you can see each of these mutations has higher nucleolar localization. And we see a cooperative effect if we further disrupt the PWWP1. as shown in the blue box here. So this suggests a cooperative effect amongst the reader domains for localization of this enzyme within the nucleus. And so, it's the concept of multivalent chromatin targeting, and that disruption of individual domains may have very specific effects. They won't completely eliminate the enzyme from chromatin, but they can have mild specific effects. And in this case, NSD2 chemical probe does not, it's not required for maintenance of global H3K36 dimethyl levels, as shown in this Western blot here. So there's still same amount of H3K36 dimethyl with increasing amounts of chemical probe. Same as the active is the same as the inactive control. And in our hands, it also had no growth suppressive effects in multiple myeloma cells. So it can modulate localization within the nucleus a little bit, but it doesn't, as far as a cancer therapeutic, it's not as promising as we and others in the field had hoped. However, over about the same period of time in the late 2010s, this new modality of drug was really starting to come into effect, and that's called targeted protein degradation. And this is a modality where a ligand to even a non-functional region of a protein can be exploited to induce the degradation of the target protein. And so back to this figure I showed for these proteins that we had made chemical probes to. We and others have now made targeted protein degraders to a number of these. And in the fields like the bromodomain, there are many targeted protein degraders to certain bromodomains, particularly the bromodomains in the BAF complexes, SMARCA2 and SMARCA4 bromodomains that are now making their way through the clinic with some pretty promising results so far. So this is one of the hottest areas of drug discovery right now. And David Nee, a grad student in my lab, followed up on the work of David Dilworth on the PWWP1, again, and further collaboration with Lindsay James's group. And we made these bifunctional molecules that were meant to bring NSD2 any isoform of NSD2 that contains the PWWP domain into close proximity with a ubiquitin E3 ligase that would trigger its polyubiquitination and then degradation through the proteasome. And that's a long story. I won't go into it, but suffice it to say, we were able to identify such a molecule. It's shown here. And it turns out that it recruits this FBXO22 complex of colon 1 complex that can polyubiquitinate NSD2 and any isoform of NSD2 that contains the PWWP1 domain. So this is a really cool type of pharmacology. It's catalytic. So because the target protein is degraded, this complex, the E3 complex is acting like as a catalyst. And so you don't need large concentrations to fully saturate all the site, because the binding site's going away. It's described as ways to drug the undruggable. It could be cell type specific. In this case, it's not, but if you were harnessing an E3 that were only expressed in certain cancers, for example, or certain cell types, then it would be a cell type specific therapeutic. And again, it's one of the hottest areas of drug discovery. So this is our entire collection of chemical probes developed by the SGC, and there's a whole cohort of also similar compounds created by other groups. One of the things we like to do is to use the entire collection as as a way to empirically probe disease cells, particularly cancer cell lines or tumor organoids, to ask the question, if we see a phenotypic effect in the case of cancer, it's usually growth suppression or apoptosis or cell viability. If we see if we see the type of effect that we're looking for, such as the dark bars here show growth suppression, then we can reasonably expect that the target of the chemical probe, in that case, is responsible for driving the growth in those cancer cells, and that this is interesting, at least an interesting target to look at in terms of that particular cancer. So you can see that there's, there only a few of these have very strong effects across multiple cancer types, but there's a lot of cases where you have certain subtypes of a given type of cancer that are vulnerable to one chemical probe. And those are interesting cases because in almost any type of cancer, any given therapeutic is not effective across all patient types. And so patient-specific vulnerabilities are very much of interest. So we've done a lot of work in this area over the years, and just a demonstration here of the usefulness of these chemical probes in cancer research and understanding epigenetic-driven pathways. So I'm going to end there and hand over. First, thank all my colleagues, especially my chemistry colleagues that helped develop some of the molecules I talked about today. All the members of the SGC, trainees and pharma partners, and my lab at Princess Margaret, all the members shown here. And then I'm going to hand over now to Riley to give the second half of the presentation, and I'll we'll answer questions at the end.
All right, thank you so much. That was fantastic. Next, we have Riley Horvath, who completed his Ph.D. of biochemistry and molecular biology in the Sadowski Lab at the University of British Columbia. During this time, he published a number of papers describing HIV-1 latency modulating mechanisms and the use of chemical tools to exploit host cell factors with potential therapeutic implications. As of September 2024, Riley has been a postdoctoral researcher in the Arrowsmith Lab at the University of Toronto, and his work aims to describe the role of H3K36 methyltransferases in small lung cell cancer progression.
Hello. So I'll just start this up here. Come on. You can give me a nod if you see that. Okay, awesome. Okay. So thanks for having me. Again, I'm Riley in the Arrowsmith Lab. I'll be talking about looking at NSD2 and its possible implication in small cell lung cancer progression. And as a disclaimer, yeah, I just did start my position in September, so the project's still pretty naive, but starting to get some fun data now. So since we're talking about NSD2, talking about histone lysine methylation most often. So here's just the lysine residue. The action of lysine methyltransferases induce mono, di, and trimethylation. And shown here are the various histone lysine methyltransferases, which are specific to certain histone lysine residues. Then counter to them, histone demethylases strip off those methyl groups. And it's important to know that in many contexts, this is not a subsequent reaction. For instance, unmethylated lysine can be trimethylated. It doesn't have to go through a stepwise process. On the right here is just looking at some commonly studied lysine residues, which are known to be methylated. And this methylation has implications in various processes, but often we look at transcriptional regulation, which in the case of methylation can be positive or negative. So here, H3K4, K36, known to be associated with activation, while K9, K27 associated with repression. So here we're looking at the K36 methyltransferases, of which there are 5. You got NSD1, 2, and 3, as well as ASH1L, which are specific to K36 dimethylation. NSD1, 2, and 3 are quite related, sharing about 70–75% sequence conservation. However, NSD2 typically generates the bulk of dimethylation in most cell types, including cancer cell lines. Then finally, we have SETD2, which is the only K36 trimethylase. And based on schematics, Cheryl talked about it a bit, but you have these proline, tryptophan, proline domains, plant homeodomains, bromodomains, which bind to chromatin and can spread the mark. So we know that K36 di and tri is associated with transcriptional activation. So here's an active gene. You see a peak of K36 dimethylation surrounding that promoter, that transcription start site, where it tapers off going into the 3' direction of the gene. Converts to dimethylation, trimethylation increases towards the end of the gene. The reason being is that SETD2 is carried along by elongating Pol2. And another important facet of K36 methylation is that it inhibits PRC2. So it prevents the spreading of repressive K27 di and trimethylation, maintaining active regions. And why do we care? So these 5 methyltransferases have been linked to a number of cancers. SETD2 is broadly thought to be a tumor suppressor, while NSD1, 2, 3, and ASH1L are correlated with oncogenesis. NSD2 in particular tends to be overexpressed or hyperactive. For instance, multiple myeloma, a translocation puts NSD2 under control of a strong promoter driving its overexpression. In acute lymphoblastic leukemia, about 15–20% of the time, there's this mutated NSD2 glutamate 1099 lysine, which increases its catalytic activity, resulting in enrichment of K36 dimethylation and is associated with poor prognostic outcome. And then non-mutationally in several cancers, breast, prostate, lung, NSD2 is just overexpressed. So recently, there's been a lot of efforts in order to drug these various methyltransferases, and we've focused a lot of that effort on targeting NSD2. So again, this is the UNC 8732, which is the NSD PROTAC. Through the characterization of this molecule, actually came up with this inactive molecule. Again, Cheryl mentioned the only difference is this moiety, which makes it a really nice molecule. Furthermore, UNC 8732 is actually inactive in this state, needs the deaminase to occur, producing this aldehyde species, which then can degrade through the ubiquitin proteasome system using FBXO22E3 ligase complex. So moreover, in the study, David Nee from the Arrowsmith lab used this molecule to examine acute lymphoblastic leukemia, where you see the degrader removes NSD2 long and short isoforms quite effectively, and in correlation, you get a decrease in dimethylation of K36 and an increase in trimethylation of K27, presumably spreading into areas that have lost K36 dimethylation. Additionally, upon depletion of NSD2, you get increased apoptosis and decreased cell viability in acute lymphoblastic leukemia. So now this is where my project takes over. We have this nice molecule, great at degrading NSD2. So can we apply it to some of the other cancers where NSD2 might drive oncogenesis? So we decided to look at small cell lung cancer. So here's just a picture of lung epithelium, various lung cells in there. Upon loss of TP53 and RB1, that can generate small cell lung cancer. So there are multiple types of lung cancers. Generally, we split it into small cell lung cancer and non-small cell lung cancer, which is lung adenocarcinoma, which is this adenocarcinoma is included in. And interestingly enough, if you take a non-small cell lung cancer and lose TP53 and RB1, it can actually transmutate into small cell lung cancer. And why we care about this disease in particular is that it's highly recalcitrant. Its average survival is dismal, being 10 months, with a 2-year survival rate of only 6%. And the one thing we do know is that NSD2 is broadly overexpressed in lung cancers of all types. Furthermore, small cell lung cancer is a neuroendocrine malignancy. So on the left here, small cell lung cancer is genetically homogeneous, loss of TP53, RB1. However, there's these various molecular subtypes defined by lineage-defining transcription factors. So there's 4 predominant subtypes: ASCL1, NEROD1, POU2F3, and this generally inflamed subtype that lacks a clear transcription factor. But looking at ASL1 and NEROD, which comprise the majority of small cell lung cancer, these are neuronal transcription factors. So when they're expressed, they bind to the promoters of neuronal genes, drive expression. And importantly, though, these subtypes can undergo molecular switching. And the switching from perhaps NEROD to PO2F3 has been shown to be a mechanism that generates resistance to chemotherapy. And interestingly, the connection comes from neuroendocrine prostate cancer. So recent studies have shown that NSD2 supports the neuroendocrine form of prostate cancer. So on top here this is an organoid model where CHGA is a marker of neuroendocrine, while VIM is a marker of non-neuroendocrine. So in the control, you see a bunch of CHGA neuroendocrine cells. You delete NSD2, there's a complete shift, near complete shift towards this non-neuroendocrine feature, which is the white staining here. And in prostate cancer, this actually lessens cell proliferation and sensitizes it to chemotherapy treatment. So the idea is perhaps in small cell lung cancer, NSD2 would have a similar role. So this is where I began my work, just looking at expression of NSD2. So in the top left here, this is just various cancer cell lines. In the blue is the RNA expression of NSD2. Green is in non-small cell lung cancer. Blue, I should say, is small cell lung cancer. And it's actually quite highly expressed comparative to the rest. B and C now are in primary tissue. So on the left, this is normal, healthy lung compared to expression in small cell lung cancer tumors. And there is a significant increase in NSD2 mRNA here. And C is a gene array looking again at normal, as well as these non-small cell lung cancers. This is lung adenocarcinoma, lung squamous cell carcinoma, and large cell neuroendocrine. And again, small cell lung cancer has the greatest expression of NSD2. Now on the right, looking at patient outcome. Here, this is just all different types of lung cancers, and I just split it right in half between patients with high and low expression of NSD2, and it's quite clear that those with high NSD2 expression have worse prognostic outlook. Then extracting just the small cell lung cancer patients from that original data, which only left me with 19, but there's still a significant decrease in overall survival outcome. Finally, in F, now looking at progression-free survival. So these patients came in, received their standard of care treatment, and then follow-up was performed to see when remission occurred. And again, remission occurs faster in the NSD2 high group than low. So collectively, NSD2 is highly expressed and associated with poor prognostic outcome in small cell lung cancer. Which brings me to what happens in small cell lung cancer cell lines when we get rid of NSD2. So these are 3 different small cell lung cancer cell lines for which the degrader, UNC8732, removes about 80 to 90% of NSD2 in all 3 cell lines. And with that, we get depletion of K36 dimethylation and a slight decrease in trimethylation. However, which was quite surprising, was that we see no change in K27 trimethylation, which goes against dogma. And on top right, just showing that my antibody actually works. So what we see with the K27 is real. So with this, though, what are we doing to proliferation of these cell lines? With SHP77 completely unaffected here in cell proliferation as well as viability. H2286, you get modest decrease in proliferation, but it takes a long time. And yeah, it's not that great with no effect on viability. H446, the decrease in proliferation is obvious quicker, but again, relatively modest. So at this point, there's no result that's jumping out. So I started to think, what are these other methyltransferases doing? So again, there are 4 dimethyltransferases. NSD2 does the bulk. But is it possible that in the absence of the NSD2, these other methyltransferases compensate to some effect and allow proliferation to continue normally, relatively normally? So just looking at expression again in small cell lung cancer, these blue dots, NSD1, 3, and ASH1L have the very high expression of their mRNA in the small cell lung cancer. SETD2 is relatively high compared to the rest as well. And in all 4 cases, small cell lung cancer has greater expression than non-small cell lung cancer. So perhaps there's something to this. And one way to look at this, I could do a systematic approach, knocking out, depleting one at a time. But it's been shown that this H3.3 histone variant with a lysine 36 to methionine mutation limits deposition of all K36 methylation, so effectively antagonizing all 5 of those methyltransferases at once. So in this study in mouse mesenchymal stem cells, they did a systematic knockout of each one of the 5 methyltransferases. This is in mouse, and they found that NSD1 was responsible for the bulk of dimethylation, while NSD2 also contributed. ASH1L, NSD3 had no effect. And then looking at K36 tri, SETD2, most predominant, which is known. And similar to my results, NSD2 has some effect. But again, you use this histone variant, this oncofistone, sort of mimics depletion of all 5 of them. So in this shotgun approach, I applied it to my small cell lung cancer cell lines using a lentiviral transduced tetracycline on H3.3 HA tag. So here in the presence of doxycycline, the wild type is expressed. Here's the mutant version, and at 4 or 7 days. And what we see is depletion of mono, di, and tri. Again, no effect on K27 tri, and this holds through for all 3 cell lines. So it was a nice way to get rid of the K36 marks. Now looking at proliferation, here's SHP77, which NSD2 had no effect on. Start to see a significant decrease in cellular proliferation with that mutated H3.3 K36 methionine. No effect on viability though. And then H446, which NSD2 loss moderately impeded, we're now seeing significant loss of cellular proliferation, and over time do see an effect on viability. DMS114 is similar, really stunts cell growth, start to see loss in viability. So this didn't match up with the NSD2 degradation very well. So the question is, which K36 methyltransferase or methyltransferases are leading to this result? So then looking at these again, most people point to NSD1 as sort of a major player in K36 dimethylation. NSD3, its in vivo activity is not well defined. ASH1L actually does target a subset of genes, usually developmental, with K36 di, and SETD2, the trimethylase that is predominantly thought of as a tumor suppressor. So we don't have much to work on, but I had this progression-free survival data And although NSD2 is the most significant determinant here of remission, we do see significance with NSD1 as well as ASH1L. So this pointed my nose towards these two proteins, of which there is a good inhibitor of ASH1L. So a couple years back, this group discovered this AS99 molecule, which they found to bind the set domain of ASH1L and inhibit its methyltransferase activity. And shown on the right, it's quite a specific drug. So this is an in vitro panel with isolated nucleosomes and a high concentration of AS99. So you complete inhibition of ASH1L, but really limited effects on other methyltransferases. Shown here, NSD1, 2, and 3 are completely unaffected. SETD2 is also unaffected at this extremely high concentration. So what does this drug do to my cancer cell lines, my small cell lung cancer lines? So now, SHP77, within 3 days, you see a dose-dependent restriction in proliferation, while no effect on viability. H446, a stronger dose-dependent repression, 10 micromolar is cytotoxic. DMS114, it's very similar, 10 micromolar being cytotoxic. H196, 10 micromolar cytotoxic, and interestingly, DMS53, which is completely resistant at these concentrations. And what's fascinating is that the ASH1L inhibitors seem to phenocopy expression of the K36 methionine mutant histone. So again, SHP77 was relatively robust to expression of the oncohistone, and again, pretty robust to AS99 treatment. Whereas H446 and DMS114 are much more sensitive to the K36M, and they're also much more sensitive to the inhibitor of ASH1L. And why this is interesting is because previous groups have looked at H3.3 K36M, and actually in chondroblastomas, this is a science paper over here, where this histone is actually mutated and quite frequently in chondroblastomas and drives oncogenesis. And they came down on NSD2, which is MMSET, and SETD2 as the reason why this mutant drives cancer progression. Here, Nature Communication paper pointing at K36M having the same effect as NSD2 depletion in adipose tissue development. And a more recent PNOS paper, which pointed to knockout of NSD1 and 2 is recapitulating the H3.3 K36 methionine. So to me, this is very interesting that I'm seeing ASH1L as perhaps mimicking most closely to K36M. The reason being is ASH1L is typically thought to only methylate and regulate a small subset of genes. So in the context of small cell lung cancer, if we can affect a small subset of genes and actually inhibit proliferation, it would be much more specific than any other treatment, perhaps. So with that, my conclusions found that NSD2 does not broadly regulate small cell lung cancer, but that doesn't mean it doesn't have an effect in its growth. We do see a high level of NSD2 and poor prognostic outcome. So does it have another role in any other oncogenic program, such as metastases, drug resistance, genome instability, or whatever? But I'm really interested in jumping onto this ASH1L as a potential driver of small cell lung cancer. So immediately I need to confirm the drug experiments with some genetic tools, which I'm setting up my CRISPR cell lines now, and then which genes are being affected. Hopefully it will do some RNA-seq and see a small subset, and also confirm that with the dimethylation of K36. And another interesting angle then will be how are some small cell lung cancers resistant to ASH1 inhibition? So I randomly picked 5 cell lines, and only one of those was resistance. So there's probably more, and I could probably glean some information from their transcriptomes to understand that further. And then finally, here at the Princess Margaret Living Biobank, they actually have developed a number 29 small cell lung cancer organoids from patient samples. So hopefully in the next little while, we'll start getting those over, get culturing, and repeat some of the experiments using a better model. So with that, I'm still relatively new, but thanks to the Arrowsmith lab, as I get to know more and more people, as well as the Structural Genomics Consortium, with Cheryl and Dahlia really helping me get this project going, and Shilly helping with the cloning. So with that, thank you for your time. And I guess we'll take questions as a team here.
Thank you so much for that great presentation. I have a quick question before I go to the Q&A. As you were sort of speaking, I was wondering, like, is there kind of a thought that treatment might be best if, you know, you sort of start by combining a few of these things, or is it really to say, no, we have to figure out what the subtype is and then pick the right one? Like,
So I don't have enough data, but it looks like when I combine the NSD2 degrader with the ASH1L inhibitor, it has more of an inhibitory effect on proliferation. So yeah, there's definitely a route there, similar with drug resistance for small cell lung cancer, chemotherapy of cisplatin and etoposide. These are DNA damage inducers. There's an angle where you can combine that with these epigenetic inhibitors.
mhm, yeah. All right. Okay, so going over to the Q&A, the first question is, given that NSD2's methyltransferase activity is well studied, how critical is its reader domain in driving oncogenic function? Could selectively targeting the reader domain rather than catalytic inhibition offer a more precise therapeutic strategy with fewer off-targets?
Yeah, I can answer that. I mean, that's kind of what we tried to do with the first generation chemical probe, just to the PWWP domain. At least that reader domain didn't have any effect on you know, oncogenic properties or drivers. That's why we resorted to turning it into a degrader. Not to say, well, one would have to test all the other reader domains. There's a lot of them, right? So yeah.
Okay, thank you. Are there specific oncogenic pathways that NSD2 interacts with in small cell lung cancer? And do these differ from other cancers where NSD2 is implicated?
Riley?
Yeah, so that's where you're starting to get into. So where you don't see the proliferation effects of NSD2 degradation, however, in samples from patients, you do see correlations with expression, prognostic outlook, and that implicates in itself that although division of the cancer itself might not be the important role that NSD2 plays, there are perhaps these other programs which I'll need to start investigating, probably need some RNA-seq information or anything just to look at metastases, any implications in DNA damage response, all of that. But I have not gotten there.
Okay. And then the next question is kind of technical. So you're being praised for how beautiful your Western blots are. And they've asked, do you use antibodies with different hosts to detect total H3 and the H3K36 modifications, or do you strip the membranes and reprobe them?
Different hosts.
Right.
So in my PhD, I used chemiluminescence where stripping worked nicely. With this Lycor fluorescence, stripping's not so nice. So different hosts.
Okay. All right. Does UNC 8732 have any effects on NSD2? Is it just a probe, or is it capable of inhibiting its binding with other proteins or its activity?
Right. Well, it's a degrader, so it gets rid of the protein. It induces proteasomal degradation of the protein. It's the compound binding site is the PWWP domain, so that does disrupt interaction with histones, but then the protein's degraded, so it's that the degradation trumps any other effects. Yep.
Right. Yeah. Okay. So the next question is, Reader domains play a crucial role in interpreting epigenetic marks, but many have structural redundancy and overlapping functions. How do you envision developing highly selective degraders that can distinguish between closely related reader domains without disrupting broader chromatin regulation?
Yeah, we've shown, for example, most of the bromodomain family has selective inhibitors to each one of them have been developed over time. Not all the methyl reader domains have been targeted so far, but the PWWP, the chemical probe that we developed is exquisitely selective for NSD2 over all the other PWPs in the cell. So even though they have at the protein fold level, the proteins look similar. The amino acids lining both the peptide binding groove and the methylation binding pocket are not all the same, except for the aromatic cage. So there's opportunities to design selectivity into the chemical probes.
Right. Yeah. Okay. And then there's a couple of questions about, I think for Riley, like, why do you think that you don't see an effect on the levels of H3K27 trimethylation after the inhibition? Like,
So,
do you have ideas for why?
yeah. So in the context of small cell lung cancer, you got NSD2 is overexpressed or expressed at a high level. You also have EZH2 expressed at a high level. So the thought is that the overexpression of NSD2 generates K36 methylation, which keeps out PRC2 out of certain domains, but that focuses the PRC2 into its particular domains and hypermethylates, hyper K induces K27 di and tri. Perhaps upon loss of NSD2 and depletion of K36 methylation, you might get a spreading from from those zones. However, you're also probably going to lose the hypermethylation at zones that were restricted to EZH2 is my best thought at the moment.
Right. So it's kind of already really high and you're just maybe seeing a redistribution, but not
Yes.
an actual change in the absolute amounts, right?
Mhm.
Do you have plans to do chip on some of these to look at those
Yes. Yeah.
locations? Yeah.
In both, if pursuing NSD2 as well as ASH1L, both need some chip-seq experiments here.
Yeah. Yeah. All right. Well, I think we've done super well for timing because we're just coming up on the hour. And I think that that's the end of the questions that were in the chat. So thank you both for your really fantastic and inspiring talks today. And I have a, new interest in thinking about how to drug some things that I thought were undruggable. So, maybe we'll see where that can take us. But yeah, thank you both, and have a great rest of the Friday, everyone, and a great weekend. Thank you.